Bismillahirrahmanirrahim. We start another paper 1 to November 23, A-Level Biology in the first video. As we all know, this paper is for 1 hour 15 minutes. The timing has increased since the change in syllabus in 2022. So you have more time to do these questions now as previously as compared to 1 hour papers. Question number 1. Which combination of lenses for a light microscope will give the greatest magnification? So this is 500, 100 into 5. This is 40, 400. And 15, 40, 600, and 15 into 100 is 1500. So the answer is D, the highest magnification. That is, in fact, the truth is that the highest useful magnification is only 1500 times for a light microscope. After that, even if you magnify it, it is not any useful. It just becomes blurred. The diagram shows an electron micrograph of a virus particle in a human nucleus. So you measure it. It is about nearly 3.6 millimeter. So that means it was 3600. So you multiply it by 1 millimeter is equal to 1000 micrometers. So it will be 3600 micrometer, because 3.6 into 1000 is 3600 micrometer. And then you add another 3 zeros to it, and this would be nanometers, and then you divide it by 2000, 24,000, which was the magnification here, and this is equal to 150, and that is why the answer was D, 1.5 into 10 raised to the 2 nanometers. You know how you do that. I hope you know that. I've also learned it after a very long time. You move the decimal 2 places. So the decimal has moved 2 places, so this is how you do it, 1.5 into 10 raised to the power 2. Now, question number 3. What is correct about the synthesis and release of the glycoprotein mucin in the goblet cells? The protein is produced on the ribosome and a carbohydrate chain is added in the Golgi. Correct. The glycoproteins are packed into vesicles in the Golgi body forming lysosomes. No, it will be a vesicle containing the mucus. The mucus has to be thrown out of the cells. Lysosomes contain hydrolytic enzymes and they work inside the cell to get rid of the worn out organelles. Number 3, secretory vesicles containing the glycoprotein move from the Golgi and fuse with the cell surface. Yes, that is correct. So it is 1 and 3 only. So that is why the answer to question number 3 is B. This was wrong. You have to understand why this was wrong. The lysosome thingy is wrong. It is talking about the synthesis and release of the mucin in the goblet cells. Mucin is a glycoprotein. So it was going to be that works only inside the cell. Here we are throwing the mucus out of the cell into the trachea. Then what are functions of microtubules? Question number 4. Allowing movement of cilia and a bronchus, attachment of centromere to metaphase, moving secretory vesicles around a cell. Yes, all 3 of them. The answer is A. Please understand microtubules and the function of microtubules is new in the syllabus since 2022, so you need to read it from the book. Then question number 5. Which organelles contain nucleic acids? Golgi body does not contain, mitochondria does contain DNA. Nucleic acid means DNA or RNA. So wherever there is DNA or RNA, there will be ribosomes, and ribosomes contain RNA. So mitochondria and chloroplast, that is why the answer is D. Then coming to question number 6. Question number 6. Some features of cells are listed: cell wall, cell membrane, ribosomes. Which features can be found in plant cells and in prokaryotic cells? Well, yes, both of them would have a cell wall, a cell membrane, and a ribosomes. So that is why the answer is A. Then question number 7, which bonds are present in all viruses? All viruses have a protein coat. All viruses have either a DNA or an RNA strand. So the DNA or the RNA will have a phosphodiester bond. The protein coat will have peptide bonds because it will be, and number 3, there could be a peptide bond is a covalent bond. So that is also correct. So that is why the answer is A. All 3 are correct. And question number 8. Student carried out the Benedict's test on 4 different concentrations of glucose solution and then recorded the time taken for the first appearance of a color change, which is the end point. The student found it difficult to identify the first appearance of a color change and consistently timed each solution for 2 seconds after the color change. Say it was 10 seconds, he said 12 seconds. If it was 8 seconds, he said 10 seconds. So, and each solution for 2 seconds after the color change first appeared. This introduced a source of error into the experiment. Yes, everything was plus 2. So if the answer was 8, it was 10. If the answer was 6, it was 8. If the answer was 12, it was 14. Which statements about this error are correct? The effect of the error will be reduced if the student performs 3 repeats at each concentration of glucose. Well, that doesn't matter. But every time they're going to add 2. The error will prevent the student from identifying which solution has the highest concentration of glucose. No. The error is systematic as the student consistently timed each solution for 2 seconds after the end point. That is why the answer is D, 3 only. That was the only correct thing which we was talking about. It was a systematic error. Then coming to question number 9. Which statements about amylopectin and glycogen are correct? Amylopectin and glycogen contain 1,4-glycosidic bonds. Amylopectin contains beta glucose. Sorry, beta glucose is only in cellulose. You must know that. B, C, A, B, C, D. Beta glucose is only in cellulose. That's why I've told you to remember the mnemonic for it. So this is wrong. Glycogen contains more 1,6 than amylopectin. Yes, both are branched. Amylopectin and glycogen both are branched. Glycogen contains more 1,6. So that is the answer is 1 and 3. Amylopectin and glycogen both are branched, 1,4 and 1,6 bonds, but glycogen has more because it has to be more compact. It's a storage molecule. Similarly, amylopectin is part of the starch molecule, which is also a storage molecule. Question number 10. How many statements about fatty acids are correct? How many statements? Fatty acids are either saturated or unsaturated. Fatty acids always have at least 2 double bonds. Long chains of fatty acids are very good energy stores. Fatty acids are insoluble in water and do not affect the water potential of the cell. So as you can see, one of these is incorrect. Always have at least 2 double bonds. No, that is wrong. So 3 are correct. So answer is 3. The rest are all. You've got to read up more on biological molecules and the structure of fatty acids. Question number 11. The diagram represents an amino acid. R represents a variable side chain. This can be variable, can be H, can be CH2, can be anything, CH3. What is not a possible side chain? You see, it has to bond with the one carbon, so it has to have a, which is not a possible side chain. You can see in all these, this is the only one which cannot bond, which is not a possible side chain. Question number 12. The graph shows the effect of an enzyme on a reaction. Which row identifies X, Y, and Z? X, Y, and Z. So X, the 3 possible answers are a catalyzed reaction, uncatalyzed, energy lost by the product. Now, what is energy lost by the product? You know, this was the energy, say it was 100. Now the products are at, say, 20. So energy lost is 80. So that is why the answer is D. X is the uncatalyzed reaction. You see, this is the uncatalyzed reaction. And then this has lowered the activation energy. So why is the catalyzed reaction? Overall energy released during the reaction. This is the overall energy released during the reaction. Now, question number 13. The graph shows the results of 2 experiments on the effect of increasing substrate concentration on the rate of enzyme. So we've got substrate concentration here, and we've got the initial rate of reaction on the y-axis. One experiment was at a high concentration of enzymes. So we had 100 of these, 100 enzymes, and on another experiment we had only 50 of the active site. Sorry, I'm not made the active site exactly the same. So these were 100 active sites, and here there were only 50 active sites. One experiment was at a high concentration of enzyme, and the second was at a low concentration of the enzyme. All other variables were standardized. What would limit the initial rate of reaction at point X? You see, if this graph wasn't given, then we would say substrate concentration. But because this graph is given, so at this point you can see the rate is increased when the enzyme is more. That is why the answer is A, enzyme concentration. So question number 13, the answer is A, enzyme concentration is the limiting factor. In this case, in which they have given you two graphs, if they had not given you this graph and only given you this graph, then at X substrate concentration is the limiting factor. Please understand this question. It's a very interesting question. Question number 14, which statements about the Michaelis-Menten constant Km of an enzyme is correct? At the Km value, half the active site of the enzyme should be occupied by the substrate. Yeah, well, you're right. That is correct. At Km value, half the active site. So this is the Vmax. And then you calculate the half of it, and that is the Km. So at the Km value, half the active sites of the enzyme should be occupied by the substrate because substrate is limiting. You increase the substrate, more the rate of reaction. So 1 is correct. 2 Km represents the substrate concentration at which the enzyme is working at half the maximum rate. Yes, that is correct as well. The lower the Km, the lower the affinity. This is wrong. It is the lower the Km, higher the affinity. It's a sort of inverse. When an enzyme has a high Km value, the enzyme reaction will proceed very slowly. Yes, that's correct. So 14B is the answer. 1, 2, and 4 are correct. 3 is wrong. Which component of cell membrane helps to reduce fluidity of the phospholipids at high temperatures? Is the cholesterol. We say cholesterol regulates fluidity. Cholesterol regulates fluidity. Question number 16. Plant cells were left for 50 minutes in three different sugar solutions, 10%, 5%, and 1%. The water potential, the cytoplasm of the three cells was the same at the start of the experiment. The diagram shows the appearance. What has this happened? Water has moved out. So that means what would be the case? When will water move out of a cell? When it is placed in a the inside has a higher water potential and the outside has a lower water potential. Question number 16. Plant cells were left for 50 minutes in three different sugar solutions, 10, 5, and 1%. The water potential in the cytoplasm of three cells was the same at the start of the experiment. The diagram shows the appearance of the cell after 50 minutes. Now, cell R is plasmodized, so water is moved out. Cell Q, water is moved down, it's become fully turgid and it's really become a little bigger than the rest of the two cells. Cell P is fine, but the vacuole is smaller than cell Q. Now, which conclusions is correct? Cell P has the same concentration of sugar inside and outside. Cell P, same concentration inside and outside. How do we figure that out? Because cell Q is fully turgid. Cell Q is flaccid. Wrong. Now, cell Q was placed in the 1% solution because it was a dilute solution, so water has moved in and the vacuole has, become fully turgid. Vacuole is much bigger than cell P. The sugar solution outside the R is less negative. Now, I've done that. I've shown that to you. That's a wrong statement. If it's less negative, it says the sugar solution outside is less negative. It's -5 and inside is -10. -5 means 5 grams sugar in this. 10 means 10 grams. I've told you this in my videos. -5 minus 10. -5 means it's a dilute solution. It's got 5 grams of solute in it. 10 means it has got more solute, so less free water molecules. Here there are going to be more free water molecules in a dilute solution. So that is why D is wrong. Question number 17. The graph shows the effect of increasing the side length of agar cubes on the surface area and the volume. So I give an example of 1 centimeter and 2 centimeters. So this was surface area was 6, volume was 1. Then the surface area increased to 24, 4 times. But the volume increased by 8. So 1 has to be volume. As you increase the side length, the volume increases more. So that is why the answer is C. The volume increases, the surface area is line 2, and the surface area to volume ratio decreases because this is 3:1. 1 centimeter was 6:1. 2 centimeter is 3:1. So the surface area to volume ratio has decreased. I've given an example to explain this to you. I hope you've understood this. A human embryo consists of 12 cells. At the start of day 1, the cells divide by mitosis for 4 complete days at a rate of 1 division every 32 hours. So 4 days means 24 hours in a day is 96 hours. 4 days. 24 hours in 1 day. In 4 days there will be 96 hours. So 96 divided by 32 is 3 divisions. The first division 12 becomes 24. In the second division 24 becomes 48. In the third division 48 becomes 96. Which diagrams show roles of mitosis? Roles of mitosis were what? Growth, repair, replacement, asexual reproduction. So the first one you can see this one, then the second, 3 and 4. I hope you've done this paper before, and then you go to 1, 2, 3 and 4. Now the answer is A. Why is the answer A? Have you ever thought of this? Now all of them show growth. All of them show growth. Something has happened. In all of them there's growth, replacement, repair, asexual reproduction. Now don't confuse this. Yes, this is meiosis also taking place, but there's also growth. Fruit formation. But we're not talking about the meiotic part, which is of course not in your syllabus at this point in the AS level. Now question number 20. Which events are part of the mitotic cell cycle? So there's an entire cell cycle we are talking about, just not mitosis. It did say which events are part of mitosis. It says which events are part of the mitotic cell cycle. So it will be all three of them: interphase, anaphase, cytokinesis. There's a question me reading. I also got this a little confused. I said, about how could that be? But then I realized the wordings are mitotic cell cycle. It didn't say mitosis. It says mitotic cell cycle, the entire cell cycle. Question number 21. Which statement correctly describes a feature of a DNA molecule? It has two parallel strands. No, they're anti-parallel. It has complementary bases are held together by 3. Not all 3 hydrogen. A and T has 2. C and G has 3. Yes, C is correct. It has phosphodiester bonds that are formed using DNA ligase. But why is D wrong? It has a backbone of phosphate molecules and nucleotides. No, it's a sugar and a phosphate, which makes the sugar phosphate backbone. Now, coming to question number 22, which diagram correctly shows the replication of DNA? Now, there are two things that you've got to consider. Number one is the direction of replication. That's fine, there's an arrow, but the S1 is the leading strand. Now, you've got to understand that if S1 is the leading strand, then it has to be, the leading strand is which one? Now, why is the answer C? Why is the answer C? Because if you look at it, S1 it says is the leading strand. So S1 is always in a 5-3 direction, and it has to be the 3 here. So this is the 5-3 direction. Now in all these this was wrong. This was wrong here. Here it is S2 that was wrong. Here it was S2 that was wrong. S1 is the leading strand. So S1 is the leading strand, but the leading strand is from towards the replication fork. It's in the 5-3 direction. So this is a 3. So this has to be the 5-3 direction. And this is the 5. So this will be the 5-3 direction. Now I'm just going to revise this with you. You see the leading strand is that has to start, the original parental strand has to be 3. So this is the 5-3 direction. The lagging strand is always, you see it starts from the replication fork and then it progresses like this. So these are the tiny pieces which are called Okazaki fragments. And then it will be in 5-3 direction. So this has to be the 5 end. And this has to be the 3 end. So please try to pause the video here, have a look at this and try to understand what do we mean by leading strand and the lagging strand. So leading strand would start from the 3 parental strand. So this would be the 3 parental strand. And then this would be the leading strand. So it is the 5-3 direction. This would be the leading strand. And of course the lagging strand would be in this direction. Because this is the 5. So this has to be the strand, new strand has to have 3 here. And it's going to start here 5. Question number 23. The RNA codon wheel is a tool used to find which amino acid would be translated from an mRNA sequence. You can see the use in it. So this is an RNA codon and it's called an RNA codon wheel. Codon position 1 is in the center of the wheel. So codon position 1 is in the center of the wheel. Then codon position 2 is in the middle of the wheel. And then codon position 3 is near the edge of the wheel. So this is the one which is the codon position 3. The three letters on the outside edge of the wheel identify the amino acid. So like pro, his, grin, garg, arg, met, thr, asn, lys, scr. These are the amino acids. We've talked about this many times. I do this a lot in school. The diagram shows a section of DNA coding for one amino acid. The template strand is outlined in a box and the DNA base sequence is read in the 5'3" direction. So now, as this is a double ring and it has two bonds, so that has to be adenine. So it is A. And then has to be guanine. Why? Because it's a double ring, adenine and guanine are purines. So this has to be cytosine. Now this has to be again guanine. Why? Because it's got three hydrogen bonds. 1, 2, 3 hydrogen bonds. So this has to be guanine and this has to be cytosine again. So the DNA is read in the 5'3" direction. This is the 5' end, 5' end, and this is the 3' end. We do it from this 1, 1, 2, 3, 4, 5 end. So what is the amino acid is coded for? So you see, if it is ACC, then the RNA is UGG. Let's look at that. UGG. This is amino acid named TRP. So the answer is C, TRP. Let's look at it again. UGG. This is the one that I'm talking about. So UGG is this. Let me just rub it off again and show it to you. So this is U, this is G, and then this is G, and this is TRP. Question number 24. 4 students sketch diagrams of the apoplast, which is through the cell wall, and the symplast is through cytoplasm to cytoplasm. Which sketch is the most accurate diagram of the two pathways? So they've given you different ones. You know, apoplast is along the cell wall, but it doesn't go into the cytoplasm. So this is wrong. The answer is D. Now, why are the others wrong? This is what I want you to see. Why are the others wrong? You see, Casparian strip blocks the apoplast pathway. So this is wrong here. The Casparian strip will block the apoplast pathway. So this is wrong. Why is this wrong? I want you to know that. B, why is B wrong? Again, Casparian strip and the apoplast pathway seen going through it. This is again wrong. And moreover, it did say this was the apoplast pathway. This is the symplast pathway which is going along the wall. So that is wrong. Symplast pathway is going here and then it's also being blocked. Symplast pathway is not blocked. Why is A wrong? Apoplast is not through cytoplasm to cytoplasm. It is along the cell wall or through the cell wall. So that is why these are wrong and answer is D. Question number 25. The photomicrograph shows some xerophytic adaptations in leaf tissues. You can say one is the trichomes. Two is a small sort of a space where the water vapors will evaporate and stay there. And they've got the sunken stomata. And three is the outer cells of the cortex. Which row shows the correct functions of structures 1, 2, and 3? 1, 2, and 3. Why is the answer B? Reduces the water vapor concentration gradient between the inside and outside the cell. So 1 is the trichomes of the hair which trap the water vapor. And 2 is that space which is going to not allow water vapor. So the water vapor will collect there and will not be moved out. Then reduces the evaporation from the epidermis of the leaf. 1, 2, and 3 all reduce the evaporation. And traps a layer of moist air is 1 and 2. Traps a layer of moist air is 1 and 2. The space, this little indentation, is the tube where the water vapors are going to collect. And one is the trichomes, which will, of course, also do that. Question number 26. Which processes are required for the transfer of assimilates from a source cell into a phloem sieve tube? Now, what happens in a companion cell is from a source into a phloem. Hydrogen ions are pumped out, then hydrogen ions diffuse back in, bring along with them sucrose. Hydrogen ions pumped out, so active transport. Hydrogen ions move back in, facilitated diffusion, and then S sucrose will move out of the plasmodesmata and into, by simple diffusion, will move into the phloem through the plasmodesmata. So the answer is A, active transport, facilitated diffusion, and simple diffusion. Question number 27. When sucrose is loaded into the phloem, it has to travel from mesophyll to a companion cell and then into the phloem. So mesophyll cell is this one, which I have shown you in yellow. And then it is the companion cell, which I've shown you in red, and then it's the phloem, which I've shown you in green. Now, what you have to understand is that the mesophyll cell can have less, but it is going to be actively transported into the companion cell. So the companion cell always has to have more than the phloem. So the answer is B. Why is the answer B? Because in the mesophyll cell it's 5. In the companion cell is 14, but it's active transport into the companion cell, and then here it is 10. So movement from the companion cell to the cell is down the diffusion gradient. Here it is active transport. So the answer is B. Now coming to question number 28. Which feature of transport in plants is correct for xylem and phloem? It is correct for xylem and phloem. It occurs by mass flow. It occurs by mass flow. Source to sink is only in the phloem, so it can't be in the xylem. In the xylem, in the phloem, it is bidirectional. It is passive. It's not passive everywhere. It's a pressure is created. It's not a passive process. A change in hydrostatic pressure results in the movement in the phloem. So basically, it's both by mass flow, means large volumes of it travel at a certain point in time. Just like you open a tap in your bathroom, and suddenly a whole lot of water flows out of the tap, and you have a big tank at the top of your building or the top of your house. So that is called mass flow. Alhamdulillah, that completes this part of the video 1, and we'll continue the rest of the questions. We'll continue in the next video. Thank you very much. In this video, we have completed question 1 to question 28. The rest will be in the next video.